So good evening all of you and welcome back to Excelsure Academy. So today we are going to take protein sequences and evolution. This is about how the sequence of the protein will tell you whether the organisms are related or not. Okay, so this will tell you about whether the organism is like from which origin, like whether it is 10 years old, 20 years old, or 1 million years old. So everything this can be done if you can build a tree which will tell you about the lineages. Okay, so from your ancestor, and of course the ancestor of each living being is the prokaryotes. Okay, so from that ancestor till now, how many proteins and in what way they have been change will be studied using the certain bioinfo matrices are there. So we use certain bioinfo matrices of which today I'm going to discuss about one. So this will tell you how exactly the evolutions can be studied. Okay, so let's study about this. Okay, so each protein's function relies on its three-dimensional structure. So this we know. The function of each protein is depending on its 3D structure, and the 3D structure is depending on its primary structure. So what is the primary structure? The peptide sequence. Okay, and if suppose there is any change is there in the peptide sequence, this will affect its structure. And if suppose structure is being hampered, it means what? It is going to affect the function also. So thus the biochemical information conveyed by a protein sequence is in principle limited only by our own understanding of structural and functional principle. So what does it mean? It means that whatever technology or latest understanding we have, it is all depending on the structure and functional principles of the protein. Okay, now the protein sequences also tell us how the proteins are evolved and ultimately how life evolved on this planet. So every time, whether it is any lab, whether you're talking about the cancer or any virology lab is there, every time there will be one scientist who will be very much interested in understanding how that particular protein or particular virus or particular organism has been evolved. That is what made them to exist on this planet. Okay, so there will be some person who will be interested in this. I'll tell you, there is one article is there, origin of life. Okay, so we think about origin of life means talking about the Miller's experiment or Oparin Halden soup theory, right? We all think about that, like first microbiomes or coazawates came. But there is this person who have studied origin of life using only maths formula, and believe me, it was so difficult to understand. I didn't understand anything. If you want, I can share also, but it was like beyond my understanding. I left the seminar in between itself. Anyway, so there are people, it's not only in bio, but also in physics, chemistry, and mathematical way, people are still trying to understand the origin of life. Okay. So phylogeny of the three domains of life. So this you already know that we have got the eubacteria, eukaryotes, and archaebacteria, right? So you also know that more the distance in this line, it means they are more separated. If the distance is less, it means they are closely related. Okay, so this and all we study in detail once we start about this. Now, evolutionary information is stored in protein sequences as already mentioned. For a given protein, the amino acid residues are essential for the activity of the protein and are they conserved over evolutionary time. For example, let's talk about the hemoglobin. So hemoglobin from the beginning till now, it has the conserved sequences whose function is to carry out the oxygen to various tissue. So even if there are slight changes in the amino acid sequence, but the functional part, the activity part, which actually goes and binds to the oxygen and carry out the function of transporting the oxygen, is still remaining conserved. Okay, so there are many cases where you will see that the amino acid mutation is taking place. So what do you mean by mutation over here? That in the primary structure of the protein, one amino acid has been replaced by the other, or it has been deleted, or extra has been added. But if it is not affecting its three-dimensional structure or tertiary structure, if it is not affecting, then there is no loss of function. Okay, even though at the primary level, that is the polypeptide chain has been changed because of the mutation, but the function is not changed since it is still maintaining its three-dimensional structure. Okay, please stop me if you're not getting any points. So amino acid substitutions are not always random. However, at some positions in the primary structure, the need to maintain the protein's function may mean that only particular amino acid substitution can be tolerated. So this means what? If suppose you have got certain amino acid sequence are there and it has been substituted by another one, okay, but if it is again not affecting the function of the protein, then it will be tolerated. Now when you say tolerated means what? It has been selected by the nature. So there comes the word called natural selection. This we'll study in evolution in detail, right? So when we start about ecology and evolution, so it will be tolerated. It means it has been selected in the nature since it is not having any harmful effect. If suppose it, if any mutation is resulting in the harmful to the organism, what happens then? It gets eliminated from the nature. Okay. Now, some proteins have more variable amino acid residues than the others. For these and the other reasons, the proteins can evolve at different rates. So what happens? It also depend. Now, protein evolution also depend where your organisms is actually being habitat. Like for example, I am there and you are there. If you are always, staying in very cold region and I'm always staying in the very hot region, what happens? There will be the same protein, but the function might change. Like your cells will have more of unsaturated fatty acids, right? Because you are in the cold condition. So having more unsaturated fatty acid, so the proteins, the enzymes which are responsible to carry out that synthesis of unsaturated fatty acid, you will have more. Whereas in case of mine, it will be less, right? So it is the same protein, but it will have the different expression or different amino acid sequences now, just because they are present in the different habitat. So in this way, protein gets evolved. Now, another complicating factor in tracing evolutionary history is the lateral gene transfer. So what do you mean by lateral gene transfer? So in case of bacteria, you know that they form the pili, right? And then they transfer the gene from here to here. Conjugation, you must have heard of. Have you heard of this?
Yes, ma'am.
So
Conjugation, yes, ma'am.
yes, conjugation. Okay. So in other words, it is also called the lateral gene transfer. Otherwise, the transfer gene transfer means what? From the mother to the daughter. Okay. But if it is from one species to the one bacteria to the other bacteria, it is called the lateral gene transfer or the conjugation. So during this, what happens is why it is becoming complicating thing to trace the evolutionary history, because it was not initially present in this bacteria. Just because of one gene transfer, now it is also present in this. So if I do the sequence comparison, what will I see? That these two bacteria are sharing the same gene, right? Same gene sequences. But initially it wasn't, but now it is. So that's why it becomes difficult to trace the evolution of the two different bacteria. Now, this happens in the case of antibiotic resistant genes. So we know that many bacteria, just because of lateral gene transfer, the plasmid which carries the antibiotic resistance genes, they get transferred to the different other bacteria. And eventually what happens? Most of the bacteria becomes MDR. What is MDR? Multi-drug resistance. Okay, so initially it was nothing, it was normal bacteria. Just because of lateral gene transfer, taking the plasmid from different bacteria, now it has become the MDR. Now if I trace back the evolution, like if I take out their plasmid, if I take out the genome, what will I see? I'll see that they're sharing the sequence similarity, gene sequence similarity with so many bacteria, even though the shape of the bacteria, the genome, everything is differing. Just because of this similarity, because of the sharing of the plasmid containing the antibiotic resistant genes, now I have problem in tracing its history. Okay. Now, for example, let us talk about these things. The study of molecular evolution generally focuses on family of closely related proteins. So molecular evolution means you are talking about with respect to the amino acid sequence or nucleotide sequence, your gene sequence. Okay. For example, protein called EF. So these we are going to study when we talk about the transcription translation. So there we are going to talk about the elongation factors and the factor 1 and EF2. Okay. So for example, EF1, elongation factor 1, it is involved in the synthesis of proteins in all the eukaryotes. And instead of EF1, you have got EF2 in the case of bacteria. So you see that there are similarities in the sequence and the function of both EF1 and EF2. Okay, so when you see that in different species it is present but having the same function, then the proteins are called homologs. Okay, same protein having the, same function, proteins are present on the different organism, it is called homologs or homologous proteins. If two proteins within the family, that is two homologs, are present in the same species, then it is called paralogs. If they are present in different species, then it is called orthologs. Is this clear? If you have a protein of similar function, then it is homologs. If it is present in the same species, like within the same organism, then it is called paralogs. If it is present in different species, it is called orthologs. Okay, you need to remember because this is quite confusing. Now, let us see this example. So, what does this example says about? So, we are going to talk about this. Now, you have got the species A and species B. Okay, again, gene 1 and function 1. So, one gene hashtag is A. So, you are going to have this function 1. So, what do you mean by that? This gene 1 and gene 1 are same. Okay, which is giving you the function 1 and function 1. So, it means what? Two proteins having the same function present in different species, it is called homologs. Okay, now if it is the now suppose it gets duplication, so gene duplication happens many a times. Okay, now gene duplication is happening in one of the gene is getting mutated. Now what will happen? It is having the one gene, one function, gene 2 functions are there. Now since they are the two proteins which were belonging to the same but present in the same species, it becomes the paralogs. Okay, so if you read, it is given the same thing what we have just discussed now. Now homologous protein, so we use powerful computer programs to find the evolutionary relationship. Now why do we need algorithms? Because see, it's not easy for us to sit with the sequences. You know that sometimes amino acids will be around thousands and more than thousands of amino acid sequences will be there. If you want to compare with the other sequence, you cannot sit with, like, you know, A to A, G to G, H to H, I to I. You cannot sit and compare like using your hand and only your paper and pencil. So we need computer programs. And suppose sometimes you want to compare among the 100 organisms, then you cannot sit, right? For one and two species you might be able to do, you know, sitting for at least 2-3 days you might be able to complete. But what about, suppose as a scientist and I'm asking you to compare between the thousands of proteins from different species, can you do it then in 1 or 2 days? No, it's not possible. So that's why we need certain programs. So I'll tell you about certain programs. I don't know whether you have heard of it or not, but there are certain programs are there. It's called BLAST. So P BLAST means protein, and then if you use N means it is for the nucleotide. So if you put into the Google, you can see that it is called Basic Local Alignment Sequence Tool. Okay, so there's this site called NCBI. Go to that and you can go to, from NCBI you can go to BLAST, and in the BLAST there will be two sections given. So organism 1 or protein 1 sequence, 2 sequence, run the BLAST. What you can see, they will give you the sequence similarities. So certain percentage will be given, 19% or 2%, whatever. Okay, so just go and try. You know, you can take any protein, you can take insulin, you can take albumin, just take from any two different species and just check how much similarity they have. Okay. Now, the electronic search process can be thought of as a sliding one sequence past the other until a section with a good match is found. Okay. Now, how it is being done, this is what we are going to learn. Within this sequence alignment, a positive score is assigned for each position where the amino acid sequence in two sequences are identical. So what does it mean? It means if you have got A, T, G, C, 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 H, I, J is there. Okay. Now A, T, T, M, M, H, I, J is there. So what does it mean? What this sentence means? It means that wherever the sequence similarity is there, like this, okay, wherever they are identical, I'm gonna give them the score. Let us say some score is given 10. If suppose, now in bioinformatics you need to understand that when I say two sequences are identical, it means it has to be AATT HHIIJJ. Okay. Now, if I say they are similar, what does it mean? Here, the two words similar and being identical, it means a lot to the program. Okay. So, identical means you will get a good score. If it is similar, then it means what? It means that either either arginine or lysine or histidine is there. So positively charged amino acids can replace each other. Now, suppose you have got aspartic acid and glutamate. If these two are there, then I can say they are similar. But if suppose alanine is being replaced by the threonine, then can I say no, they are different. If I say serine is being replaced by threonine, then I can say yes, they are similar. Alanine being replaced by proline, again not similar, not identical. So what happens, the score is given maximum to the identical amino acid sequence, less to the similar, and if it is not matching, the score goes to less, I mean the score value will be less. And if suppose here G is there but here nothing is there, so what it means, it is there, something gap is there, right? So then negative value is given. Are you getting this point, or should I explain it again? Is this clear what I just said? I mean, anyways, it is going to come in further slides. I'll tell you once again.
Yes, ma'am.
If two amino acid sequences are identical, it means it has to match like this AATT. Okay, so the score will be high. If it is similar, it means instead of arginine, I have replaced with the lysine. So positive charge amino acid. Then the score will be little less. Okay, but it will be there. Now, if suppose it is valine and here it is glutamate, or no, sorry, if it is valine and over here I'm having glutamate, then it will be almost near to zero. And here if it is glycine is there and here nothing is there, a gap is there, then it becomes negative. Okay, so this is how they make the matrix, and that's why I told you to study bio you need a matrix also. Okay, so this is how the two sequence has been aligned. So it is taken, some protein is there and taken from the E. coli and the Bacillus subtilis. Okay, now can you see how exactly they have made the sequence alignment over here? See, so first what this yellow shaded region means, they are checking for the identical amino acids. Okay, so you can see that G, 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 T, F, D, S, I, right? So all are the identical. So once you have these, so you get a good score. Now, if it is not matching and gap is there, so these gaps actually reduces your score. Now, if you ask me why the score is important, it will tell you, actually it will tell you whether the sequence similarity or the sequence identical or the matching between the two sequences is worth comparing or not. So good score means yes, it is similar protein or identical protein, and if you have the less score, it means what, the proteins are different and it is being wrong if you compare those two sequences. Okay, now, of course, if a sufficient number of gaps are introduced, almost any two sequence can be brought into the alignment. And that's why the programs include the penalties. Okay, so if you have got more gaps, more penalties, and this will lower the overall alignment score. Now, with electronic trial and error method, the program selects the alignment with the optimal score that maximizes the identical amino acid residues while minimizing the introduction of gaps. Now, suppose you take insulin from one species and you take, if you take another protein, let us say gluco hexokinase, you're taking insulin hexokinase. Even though you know that both are having different function, one is acting as a peptide hormone, the other one is acting as a enzyme, right? Even though you know this, but if you run the alignment, you will see that it is not at all matching. Maybe one or two amino acids might align and they will show the similarity or the identical, but you will see that the score is going to be very less. So whenever we work in bioinformatics and the score, if the percentage of identical or similarity is 60%, then we take it as a yes. yes, the proteins are similar, but if it is less, then we don't consider. Okay. Now, so to determine how to and what scores should be assigned to these different amino acid substitutions, so Stephen Henikoff and Georgia Henikoff, they have done this preparation of the matrix. Now, how they have done this, let's see that. So even the identical residues are given scores based on how often they are replaced, and using this they have made something called BLOSSOM. It is called block substitution matrix table. Okay, so it means what? If it is having the, like for example, aspartic acid has been replaced by glutamate, or cysteine has been replaced by tryptophan, all these replacement, or the other word is called the substitution, is happening. So you need to make a matrix out of it. Now how to make that? So before that, yes, if it is called blossom 62, it means what? At least 62% of amino acid residues were identical. So what word we are using here? It is the identical. It means what? Here in blossom, in the preparation of the blossom, sometimes cysteine has been replaced by the cysteine only. Okay, so substitution does happen, but sometimes it is the same amino acid only which is coming again. Now let's see the matrix table. So this is how the matrix you prepare in the case of protein sequence similarity. Now, can you tell me, anyone can tell me what is happening over here? So one letter alphabet is nothing but your own amino acid sequences, right? Let's see one by one. So if I say A and A, it means what? Alanine, right? So if suppose alanine is there and it is getting replaced by C, 4 means what? So this yellow shaded region means what over here? It is the positive numbers. Whenever you see any positive numbers, means that they are getting substituted with the same alphabets. Guys, are you getting it? So this is your blossom 62 table. Okay, so non-identical residues were assigned scores based on how frequently they were replaced by each other amino acids. So we are talking about the substitution over here. Now positive numbers, the yellow part, it is add to the score of a particular alignment, and negative means it will be subtract from the score. Now how it is being done? Identical residues in sequences are being compared. Now how they have been compared from, one second guys, 1 second. Yes. So what I was saying, so whenever, for example, let us say methionine is there and N is there. So what does it mean? Whenever it is getting replaced by the different amino acid, so you see that the negative score has been given. So these are the number of the times the negative score means it has been replaced by the different or non-identical amino acids. Now when it is replaced by the similar amino acids, then it has been given the positive score. Are you guys getting it?
Yes, ma'am. Yes.
Okay. So when you practice, actually, you will get it. I mean, it is easy. Nobody's going to ask you to prepare the matrix over here. But nowadays, nobody asks also, because in the program itself, at the end, you will get whether it is 98% similar or 70% similar or 2% similar. So what we are interested in, this percentage only. Okay. So this is the old method, and after that, many other came. When we'll start with the bioinformatics, you will come to know about that. Okay. So now certain segments of a protein are there which are found in the organism. They are called signature sequences. Now we are always interested in the signature sequences, and in some cases they are also called the motifs. Okay, so this means what? They have got these special sequences which are always present, even if the evolution is taking place from it been more than a million years and all, but still that particular sequence is always conserved. Now in the same way, they have got the EF1 alpha and EF2. So this is present in the eukaryotes, this is present in the prokaryotes. Now both these proteins, the function is similar. The only thing is that present in the different species, right? So they have got the signature sequence of the 12 amino acids, now which are present in both. And that's why they are conserved and having the similar function. Okay, so these signature sequences are important. So let us talk about this example. So again, the same example has been taken. Now, if you see over here, identical amino acids, correct? These are the identical amino acids. Now, signature sequence means you can see over here. If you see similarity, you can find only one or two amino acids will be different, and this entire sequence will be present. Now, because of this sequence, if suppose there is a variation in this sequence, what happens? It will tell you about the evolutionary divergence. Now, what do you mean by divergence? It will tell you how far the two species have become distinct, okay, from their common ancestor. Getting the point here? So the signature sequence is present, and if suppose any variation happens in this signature sequence, it will tell you about the evolutionary divergence. Now, where evolutionary divergence means how far two species has been diverted or it became distinct from when compared with the ancestor bacteria. Okay. Now, so once you have the similarity, you can plot the tree. So this is your evolutionary tree, and the example they have taken is GrowEL family of proteins. Now, what is the function of GrowEL? It helps in the protein lysis, okay, in the case of bacteria. So they have taken this particular protein and they have made this thing. They have made the entire tree. Now, if you have to study this tree, how you're going to study? So where is your ancestor over here? Somewhere here, okay. Then slowly it starts diverging. So if I take this organism and this organism, obviously it is going to be different now because the distance is too much. So more the distance means more distance means more differences will be there, and they're evolutionary distant. Okay. Now if I take these two, it means what? They are going to be having so much of similarity. Now suppose I take these, they are also different, but they will have the much similarity. But if I compare this one with this, it is going to be totally different. Okay. So the length the length over here will tell you how far the organism species the species is going to be different from the existing one. So whenever you make a tree, you need to have a ancestor, and then you compare the existing one, how far it has been distinct. Yes. So thank you for listening. If any doubts are there, please ask me right now.
Ma'am,